Good evening and very warm welcome to all of you. It's my honor to introduce Professor Sanjay Jambhulkar from Bhabha Atomic Research Centre. Professor Jambhulkar obtained his MSc and PhD degrees in genetics from Indian Agriculture Research Institute, New Delhi. And later on, he joined Nuclear Agriculture and Biotechnology Division at Bhabha Atomic Research Centre in 91, Scientific Officer C, and from where he retired as Scientific Officer H on superannuation. And he was also the head of Mutation Breeding Section and professor in Homi Bhabha National Institute. His area of specialization is mutation breeding for genetic improvement of crop plants. He has developed almost more than 8 high-yielding varieties in mustard. Also, he was a member secretary of Food and Agriculture Committee in the Board of Research in Nuclear Sciences. He has delivered more than 100 lectures all over India on the peaceful use of nuclear energy in the field of agriculture. And he has worked on many international projects associated with the Food and Agriculture Organization and IAEA in Austria. He has been awarded the Lifetime Achievement Award in 2024 of ICAR. Also, he has worked as an expert in International Atomic Energy Agency and visited US, Austria, China, Sri Lanka, Malaysia, Vietnam, Indonesia, and Cameroon. He is a recipient of prestigious NAFA IAEA International Award in 2014 and 21. Also, he was awarded the DA Group Achievement Award in 2014, 2009 and 2007. And also he is a gold medalist of the Society of Rapeseed Mustard Research 2019 and fellow of SIMR India. Today he is going to talk about the peaceful use of nuclear energy in agriculture. I welcome Professor Jambhulkar.
Good afternoon, everybody. And first of all, I thank you for giving me this opportunity to interact with you and visit this beautiful campus. As well as I have learned some in various subjects which are really interacting with each other to promote with the end of the fruitful results, which will help students as well as the fraternity. I'm really impressed upon visiting this institute. As you can see, the title use of technology in agriculture and food, because basically we are working in the institute where the nuclear sciences is the main thing, and which has started with the kind of the physics, mathematics, chemistry, and then afterwards the engineering side, and in the biology they added. They really get a lot of application of the nuclear sciences in all those fields. We'll not go into the details of other, but I will concentrate only on the agriculture. If we think of the food security as such, looking into the various problems, like basically it's a cultivated land. If you see the urbanization and the number of the new structure which are coming, at the cost of the cultivated land, okay, which is now really shrinking down. That's the one thing. Second, everybody knows that our population is increasing like anything. And the top of that, now there is a climate change. So these are really the adverse situation for the particularly Indian situation, because our population is increasing. So we need to develop the crop plants which will be at least sufficient to feed our people in India. So that is what the main concern now as well as the coming year. So if you ask the questions whether really we are the food secure country or not, sitting into this AC room, we can say yes, we are the food secure country, because we are getting the food since morning to evening. But really, if you go to the outside and see the people's condition, and unless and until you realize it, then you feel that yes, I think there is a problem of the food security. So this is the sandwich of the thoughts between the whether it's a food secure or we are not food secure countries. So it is up to individuals' perspective. I will show you the reality after some times where we are. As I said that the population is tremendously increasing, that is 142 crores at present, and our food production is 320 million tons. Okay. So whether this is sufficient or not, I will show you another graph, and as I told that 142 million hectares, which previously it was a 152 million hectares. Now the 10 million hectares has been reduced. In the coming days, again, we don't know. So one side we have the pressure of increasing population, and another side we have the decreasing cultivated area. Under that, the question is whether we are food secure country or not.
This decreases in how many years? 152 was when?
Previously it was 152.
10 years ago or 20 years ago? Or?
Yeah, now onwards we can say near about 25 years ago. You see that? Again, it's the same thing. Coming days, how much food we require. You can see here that 2050, we need, our population will be 1.68 billions, and our food production demand will go 459 million tons. And in the present, we are near about 320, which will at the end of this particular decade, 2030, we need the 355 million tons. So this is the tremendous pressure. Pressure in the sense, as I told that, because our cultivated area is shrinking. So unless and until we increase our production per unit area, then and then there is a possibility to enhance the production. And as I told that, now if you see 218 onwards, you can see that the first black graph, that is the food demand, and second line green one is the food supply. So the gap between demand and supply, it is increasing. And it has just started. In 2018 onwards, it has started.
Pointer counter.
Yeah. So this is the gap in 2050. As I said, our food production should increase 455 million tons. So whether it's really possible to reach this stage looks very difficult. And in this stage, it will be kind of the horrible situation whether we have the sufficient food or not. And therefore, what we need is an increase in the production per unit area. Unless and until we do that, otherwise, a lot of things like loss of current production capacity and food supply. And probably we can lead or we can come to the situation where the Malthus has defined the theory. Long back he said, your population is increasing like a geometric means and your food production is like a arithmetic mean. So sometimes food production and the population, there will be the conflict and we will not able to supply the food to the people and there will be like a chaotic situation which may lead to the disease, famine, war and calamity. Hope and work for that this situation should not come to the India. Let us work for that. Okay. So far we were successful in the compensating or to supply to compensate the demand of the people, and we are in the satisfactory situation. Let the Malthus prove that you are not true in the coming days also. But sometimes if you see, If you recall the 1942 to 45 Bengal famine, where the millions of people died, who says that sitting into this AC room, we don't realize the importance of the food. But there was a situation where the millions of the people died because of the not supply of the proper food. Okay, that is the situation, and the situation was like this in the West Bengal. Again, I hope and pray that this situation should not come into India. And therefore, the great philosopher Seneca says that a hungry person listens neither to reason nor religion nor bent by any prayer. If the practical aspect, if you wanted to do, you keep yourself on the fast for the 5 days. And on the 6th day, if your mother says that, okay, you first of all go, become fresh, do the prayer, and then you eat. You will say, first of all, I want the food, then rest of the things I can do it. So hunger is such a situation that it is very difficult. You don't believe on the okay. So that is what the importance of hunger, and no people in India should go to, or no people in India should face such a situation. But present situation is like this. And if you look into the global hunger index, it is based on some kind of the parameter at the international level. They decided what is the global hunger index. And at the what particular range, if the situation is there, you are in the which stage. If your global hunger index 2034, it means this your situation is very, very serious. And at present, our global hunger index 2023 is 28.7. It means that we are not in the satisfactory position. Therefore, since starting of my lecture, I'm saying that sitting in this AC room, we don't realize what is the importance of the food. But the people who don't get, and there are the indicators. There are some of the indicators, may not be foolproof, but still it is an indicator that who says that we are healthy people. But if you analyze like a genome sequencing has done, and if you analyze your sequence, probably you may find the large number of the mutations are not suitable for your health. Okay? Looking into the number of the hospitals in India and number of the patient per day visiting to the hospital, this is the indicator that whether you are healthy or not. Okay? So this is through this at least why you are not healthy. If you compare even with the China, which is the most populous country, but the kind of the particularly since birth to the child development, if you take the various parameters, still we are lacking behind. The Planning Commission of India says that the 50% of the pregnant women of India, they are anemic. It's the Planning Commission report. The situation starts from there itself. So there we can, unless and until we get the nutritious, sufficient food, we look healthy. But still nutritional weight, if you say, we are not at all healthy. We are suffering for the one or other diseases. And the indicator is that the 50% pregnant women in India are anemic. So that's the situation. And 50% means if the situation comes, if you say the birth rate as such, then the child which is born, these are the underweight. Then the development as per the weight and the height, you see the relation. That parameters also are not fitting properly. And if the situation persists like this, you and me are here, but in the future, what kind of the situation it will be there? Whether a progeny will be healthy or not. Whether it's not, I'm saying some kind of the threat, but it is the truth. progeny which you are going to get in the future if you want a good fighter to develop our military people. So we'll have that kind of. So it's a long-term thing. But the nutrition and the food, yes, it is playing the major role for the development. Therefore, there's a big challenges on the food production. Okay? So unless and until we develop a high-yielding varieties, as I said that the area is shrinking, so your per unit yield should be high. Then and then. Now looking into the climatic situation, your varieties which are producing, it should be biotic and the abiotic resistance. There are the insect pests and diseases. There is an increasing temperature. Okay, then there is some kind of the flood. If the situation like this year, where in the Maharashtra this year it was continuously raining, and the soybean crop has more or less destroyed in most of the part of it. So suppose your expected yield is say something like a 25 million tons, which are not going to get it. So it will affect the total food industry as well as the oil industry. Then you will say that no, this year the soybean oil has gone up. So climate is also playing a very important role for that. So we should have the variety which will tolerate all these things, adverse conditions. And at the same time, your nutritional quality. As I given some of the examples, and we are comparatively now aware about our health consciousness. You know, we should have this kind of the protein, we should have this kind of the fat, we should have this kind of the carbohydrates, okay? Because of the various problems. And some of the cereals which are coming nowadays, okay? The government of India is also promoting some of the cereals for keeping. So all these aspects under the nutrition quality, it is very important. Now, there are the various methods of the crop improvement, starting from the old days our selection method, then the plant breeding, hybridization, so and so forth. If you calculate coming to the transgenic plant, coming to the CRISPR plant, coming to the tilling plant, all this, there are a large number of the techniques. Why the mutation? Now, mutation means it's a sudden heritable changes. Changes which are heritable. See, we are also facing some kind of the mutation problem in our body, but we don't realize it. But as I said that if you take the your genome sequence, probably you will come to know that no, our parents were not having this kind of the problems, but you find the mutations in our genome. So these mutations probably will lead to some kind of the diseases. Okay? So that's the mutations. Mutations happen, and mutations are generally not good for anybody. Anybody mutations are not good. But if it happens with us, it is inevitable. It is with me. If I am suffering from the cancer, it is my problem. But the percent of the, suppose that increase in the population, then but in the plant, it so happens that if it is not good, you throw it out. And you select the good mutations. So there is a chance in the plants to do the mutation breeding. Now, why the mutation breeding? I said, suppose you found during the procedure of the mutation, I found a one plant, which under the stress situation of the climate, say there is a high temperature, say 45 to 50 degrees, and I am doing my mutation breeding experiment. So I put my population for the growth, and then I found almost all plant, 99.9% are dried because of the high temperature. But there is one plant which is sustaining under the stress climate, under the high temperature. So I will select that. Okay. So in the plant system, there is a possibility of rejecting the unwanted mutation and selecting the best. So there is a chance. And like this only, you have to do the selections under the mutation. Create the stress situation for the required character, and whosoever is a survive, you select that. Whether it's a disease, insect pest, or the high temperature, or the frost tolerance, anything. As per your objective, you select the mutations and go on. Once you select that one, you are guaranteed that this plant is going to tolerate in the future. It's their progeny. Mutation is the character transfer from the one generation to the other. So their progeny will also be true. Now suppose instead I have taken the two plant, one is having the resistance susceptible. I crossed it and then in the subsequent progeny, I want to select it. But then you will have to wait for the sixth generation to become a particular plant uniform. But in this situation, the moment I got it, next generation it will be true breeding. So your rest of the period is saved. Okay? The time which I take to isolate the right recombination through the other procedure, that 4 to 5 years I have saved. And here immediately I will plan the next experiment for the particular mutation. And therefore, it becomes a quick method compared to the other technology to isolate the desired mutations. And these are all the, what I'll say, the odd situation, erratic rainfall, temperature. I have given the example of the climate change. Second is a germplasm erosion. Sometimes you must be talking with your parents or the grandparents. They say, no, previously we were having the very good material. Heat की ऐसा लगता था कि चपाती बहुत नरम हो जाती थी. It's a common thing. Nowadays the chapatis are not good, they are becoming hard. It means that there was a character in the plant, in the previous germplasm, which has been lost. And we people are so selfish. Whatever is good things you keep it, and rest of the things you throw it. And that is what happened in the germplasm. If it is not good, just reject it. What is essential, nowadays you see Government of India has honored the two ladies, where these ladies are keeping their age-old germplasm, maintaining the germplasm of the particular crop. And that has become a precious treasure for the particular crop. That yes, you take it. In the Maharashtra, there is one farmer, he grows thousands of the line of the rice. That article has come in the front line that time. It was the time maybe the 1994. So he was claiming, if you are having this disease, you eat this line. If you are having this disease, you eat this line. With the experience. Nowadays you can analyze it, what is the content of all this, but he was so intelligent. And now that germplasm being exploited now. Develop now if you see some of the sites, if you see food as a medicine. Okay? Food was not like a medicine, it was like a natural this thing. But now the terminology has come, food as a medicine. We also look, okay, whether it's a germinated moong, okay, I will eat it. If it is not germinated, then I will. So we are also health conscious. Yes, food is a medicine. But that terminology was never. Because of the suffering, our suffering, now we are looking into the food as this is what instead of taking for the long run of like Ayurveda, you take the proper food. I think you will be healthy. Which our parents and the great-grandparents, they used to say, no, Baba, you eat like this. You eat timely. You eat this much amount. Okay? But who cares nowadays? We are busy with so many things. Okay? So some of the things which have come because of our suffering. This have not come, it is not our fault. The situation has compelled us. Okay? There are so many factors, it is not one. But in the end, when we suffer from the particular disease, that time we think, no, next time onwards I will take care of. There should not be any kind of stomach upset, or there should not be suffering like this. I will take care of it. When we suffer, that time we realize, no, we will have to take care of the health. Naturally, food takes care of you. You only will have to realize what is it. Okay? There is another scientific breaking linkage. Suppose the groundnut is very big. Generally, it is said that there is less soil. But we have some of the mutations where you can have the bigger seed with the higher oil increase. So this is the breakage of the linkages between the gene. You can bring the genes together, which is having which previously we used to say, no, this is not possible. But with the mutations, yes, there is a possibility to bring the two genes together. Therefore, it is mutation not only today it's helping in our life. Since the evolution, millions of years ago, if you look into this particular plant, maize plant, it is like a teosinte, which is a maize plant. Now with the mutations and the selections and the breeding, we have come to this maize stage, okay? We are at that stage. And now from there, again now the bigger cells, okay? The first work who has done? The nature has done. Nature has changed the genome of the particular crop, and therefore we have come to the particular situation. And then using that germplasm in the hybridization program, we could able to develop the hybrid varieties. Likewise, you see the tomato. It was initially very small size, but now you can see the variety of the tomatoes with the bigger size. So again, in between this particular germplasm was lost, the small tomato. But now, again, it's a human behavior. It's not the fault of you. Now it comes as cherry tomatoes. In my field, we used to throw it. We used to not look in, we don't want this. But now, you people goes and says, no, we want a cherry tomato. He says, no, 150, 200 grams. Want? Yes, you got cherry tomato. So, it is like this. The mutation has helped us a lot since the beginning of the evolution of plants. Not only any animal or the human being, the mutation has played a very important role. And the very, very important role in the mutation breeding is the contribution of the dwarfing genes in the wheat and rice. Because of that, the green revolution in the world and India happened because of this development of these small dwarfing genes. Previously, the rice and wheat, they were very tall, but there was a problem of lodging, there was a problem of yield loss. But because of the isolation of these dwarfing genes in the wheat and rice, whatever high-yielding varieties we have developed at present, and we say the India also there is a green revolution, okay? Because of these. And these are what? These are all the spontaneous mutations. The mutations which have occurred in the nature. Now if you if you think of, had this mutation would have not occurred that time, whether the green revolution would have been taken place or not. It would have taken place, but what is required for the green revolution? Say 50 years before when the normal, I will show that slide, had this great gentleman and the scientist and Nobel Prize winner had not thought that, no, this is what the precious germplasm, this is what the precious mutation, which requires to be incorporated in the high-yielding varieties, develop high-yielding varieties, and then there is a possibility of increasing them. You see, it's a very simple work by the nature. It's a spontaneous mutation, which has revolutionized the wheat and rice cultivation. And because of that only, at present, we are getting the food. So mutation, single mutation helps like anything. You need to analyze what is that mutation, where it is required, what is the importance of it. And that is called as a SD1 rice and RST1 and 2 in the wheat. These genes are now called as the green revolution. He is a great personality, Norman Borlaug, who is a Nobel Prize winner. You see, here it's a single base pair mutations in the, it's a single base pair change. What is that? We can do the things like a CRISPR and we can isolate the mutation. Yes, we can do it. But when it was required, the nature has helped us. Yes, there is a mutation. You take this and you improve the yield. So, because of that, if you see our food production, okay, or the wheat production, just from the 20 or 25 million tons, million hectares. Now it has come to the near about 3,500 kg per hectare. Now it is still more than that. 50 quintals, 60 quintals, 70 quintals, and now the peoples are thinking for 100 quintals per hectare. So that is the exploitation again, mutation. And this is the great personality. You might have heard about Dr. Swaminathan, M.S. Swaminathan, who is responsible for the Green Revolution which happened in the India. And let us thank him to get the food. The very good example, recent example I give in the 2013 and 14, the disease resistance. In the Kenya, they observed that one of the diseases rampantly affected the wheat crop. And there was a threat all over the world that this disease will spread all over the world. And each and every government in the world has taken precautionary measure that no seed should come from the Kenya to our country. And the government of India particularly, they have said, you screen over all the varieties and germplasm and you find out that this particular disease that is called as the UG99 stem rust, you find out the resistant strain in our germplasm. And luckily we had that germplasm in our hand and they started breeding program on that. But it didn't happen, that spore didn't come to India and we are saved. Now, what is the importance of that? It was a crop. Had it been happened like a corona, our total wheat system would have gone in a big problem. Okay? And had we not been having that particular germplasm, probably for that year or the next 2 years or the next 5 years, probably would have not got the chapatis. Yeah, government of India would have brought some kind of, but that was a problem for the all over the world. So where from we'll get the wheat then? So if the disease happens, everybody should be careful, like a situation like a corona should not come for the human being or in that way or in that terms, even for the crop also. So there again the mutation breeding only it helped. So they took the 5 kg of the seed of each of the variety, they started irradiation, and out of the billions of the plant in the M2 generation, which were grown in a 10 hectares of the land, they could identify the 7 plants only, which were resistant for the UG99 stem rust, and then they have developed the variety. So that is the importance of the mutation. So if the situation is there, only mutations can. The similar things happened in the Japan. This is a PS food, okay? And if the black spot disease comes on the leaf and the plant, you will not eat that fruit. So total cultivation in the Japan, particularly for the pears, it was susceptible for the black spot disease. And there was only one plant in the gamma garden. Gamma garden is like this particular area where in the center there is a gamma source which emits the gamma rays and affects the plant. That was the old method. Okay? But in that garden they found only one branch, okay, which is having the no disease at all. They took out this and then they made a large number of the plantation out of that, and in the present year's cultivation in the Japan is out of that particular branch, which was again the mutation. Okay? So that is how they saved the PS cultivation. Another thing, now when you take the 2 particular lines, yeah.
But by doing that, you may be losing something else also. Because that particular tree has something positive.
Yeah.
It may also be carrying something negative.
Possibly. But in the given situation, what was needed is that it is resistant. Okay? And there is no option. Like a corona, no option. You have to be inside in the house itself only. And those who have ventured or something happened. So as per the situation, you find out the particular mutation, which is helping. That point of view. Otherwise also, in the rest of the life, now there is no black spot disease in the Japan. These examples I am showing in the normal breeding method. What happens when you are taken the now since independence or the before that? As I said, there is a genetic erosion. We have lost the important germplasm. And whatever germplasm was there, extensively used in the hybridization program to develop the high-yielding varieties. Extensively. So as a result, whatever high-yielding lines, you are completely exploited. And whatever we got it, yes, this can go up to the 70 quintals per hectare. But looking into the situation again, to if you go to the first slide, that the area is shrinking and the population is increasing. We need to develop a variety which will be the high yielding than the existing one. So how to come up? We have, because our germplasm is already utilized. Even though if you are going to do your extensive efforts, you are going to result into the particular yield level. This is the example in the tomato, which has appeared in the Nature Genetics. If you use the two mutations in the hybridization program, your yield, this is the standard yield. Okay? But if you use in the, this is what these are the two mutations. This is what the first line and this is what the second line. The amount of the jump or the yield using the two mutations, it's very. You want to double it. See, if you use these two particular mutations, two lines, probably you may get more than this little bit. But now, because there were two mutations, you got one hybrid which is like this and another. So there is a jump, high jump in the yield. And therefore, Bhabha Atomic Research Centre, it's our mandate to use the mutations in the, because we are working on the nuclear sciences. So using the nuclear energy for the various aspects, that is our mandate. So we worked on that, oilseeds, pulses, cereals, okay? These are the main crops on which we work, and with the various objectives. The first objective is the high yield, disease resistance, insect resistance, heat and drought tolerance, quality aspect, desirable plant types, and then the characterize these mutations. Now, how to wear? This simple example, this is our gamma cell. Previously, we were having the gamma garden. As I said, like a Japan, they have the gamma garden. So we are also having. Then you keep the plant and expose for the longer. That was the old method. But now we have the gamma chamber, where you can keep the seed and expose it for the particular period. Okay? And then the changes occurs at the chromosome and the gene level, and then you select the desirable one. And the plant material which can be used for these particular studies are the seeds, plant cutting. You can use as per the method of propagation of the particular plant. You can use either cutting or the seed or the bulbs. Okay. Okay. So this is again just for the information. These are some of the agents which are responsible for the mutations, like physical and chemical agents. So these are the various gamma particles. Generally, we goes for the gamma rays. And ultimately, in the initial generation, we get the situation like this, that we'll have to develop the lethal dose 50. Lethal dose 50 is when you take 100 seeds, grow the plant, irradiate it and grow the plant, so 50% plant will die. They are not able to tolerate that much amount. So wherever, and why the LD50? This is the dose where your expected mutations likely to happen. So therefore LD50 is very important. So upon doing all these studies, you see the effect which you get. Once you increase the dose, there will be the large the plant stature, structure, cells, everything is affected, and again, if you increase the dose, there will be the death of the plant. Okay, so what you will have to find where there is a 50% reduction of the height. So that is the LD50. One is the number of the plants that died, another is a reduction in the 50% height. So this is wheat, this is mustard, this is groundnut. Okay? And if the objective is to only enhance the germplasm, so you go for the more than LD50. So you can guess the tremendous variability. But if you want that without much effect in the plant, you are changing one or two particular characters, you stick to the LD50 or even less than that. So rest of the characters of the particular plant will remain intact and you will get the change in one or two characters. And ultimately, the results which you get, it is a product of either the chromosomal mutations, gene mutations, or the cytoplasmic mutations. These are the deletion, duplication, inversion, and the translocation, the chromosomal changes. And at the gene level, you get the transition, transversion, or the frame shift mutation. Either there will be the double strand break, single strand break, or there will be change in the bases. This is the standard procedure which you get. You feed the seed, raise the M1 plant, and from M1 plant you raise the M2. You see the number. Okay. So your M2 population should be as high as possible. Because the probability of getting the one mutation is a 1 out of 1,000, 1 out of 10,000, or 1 out of 1 lakh. That is the rate of the mutation. So you get a more number of the mutated plants. You, as I said, then the wheat, they have grown the 10 hectares of the area, and they were able to isolate only 7 plants which were the disease resistant. So the population makes a success story. What the population? The success of the getting the desirable mutation is more. Then in the next generation, M3, it will be true breeding. Or yes, you take some small trial, and then in the M5, 10. So these are the 7 years it goes to stabilize the mutation. At the end of the 3 generation, as I said, it becomes true breeding. You try to enter into the yield trial with the agricultural universities or with the Indian Council of Agricultural Research. See, this is the M2 population. You see, this is the segregation. This is just to show segregation for the yellow plant and the green plant. So these are the chlorophyll mutations. They just to show that this is how the mutation. Wherever there is a mutations in that particular plant, if you sow the plant to row progeny, they will show the segregation. Now this is like a phenotypic changes you can see. But suppose you want some biochemical changes, okay? So then you have to screen that population for, say, oil content or the fatty acid composition, protein content or the amino acid. So you have to screen that. Disease resistance, if you want to screen your M2 population in the disease nursery. High temperature, suppose. So you expose your M2 population under the high temperature condition. So the methodology and the population, if you follow strictly, with the experience, we can say that yes, you get the mutations. You have to select for it, creating the particular norm. So this is the segregation, and generally if it is a kind of the single gene segregation, it shows the Mendelian segregation number of the plants, 3:1, you get it. That is the confirmation of the mutation. So while selecting, as I said that if it is not good mutation, or there is no mutation, you reject it. If it is a negative mutation, that also you reject. And select only those mutations which are good. So fulfill your objectives. And rest of the population you can throw. The character which we want to improve through the mutation breeding. With the experience, we can say you close your eyes and think of the character. I want the mutations for this particular character. I can say that yes, you will get it. But whatever procedure has to follow, that has to be done. First of all, number of the plants and the screening technology. If you follow this thing, you are likely to get the mutations.
Sir,
Any character. Yeah.
is it possible that through like multiple mutational breeding that we lose diversity in the plant genome?
Induce the diversity in the plant.
Like we reduce the diversity.
Reduce the diversity through the mutations. No. mutations always enhances the diversity. Suppose there is one line which is a true breeding, isogenic line. You says that, okay, the one particular genome. Now you started the mutations. Gamma rays or the chemical mutagens or the any mutagenic agent, it affects at the DNA level, very high level. So you get the tremendous variability. But sometimes you see, suppose there is biochemical mutations, which you are not able to see. And the phenotypic observation, if you take, probably there will be uniformity, but tremendous for the leaf, shape, size, height, color, you get the tremendous variability. So it enhances mutation basically to strengthen your templasm, to enhance the diversity, not to reduce it. Okay. In one of our mutations, that is TPM1, initially it was isolated on the yellow seed coat in the mustard. But afterwards, we found that there is a biochemical changes. It has increased the oil content. There was a mutations for the erucic acid. Erucic acid is one of the content biochemical fatty acid. So that was the reduced, and it was a powdery mildew resistant. So the multiple mutations you can isolate within them. It strengthens your diversity. So any character, give the particular situation, screening method and the population, you are likely to get the mutations. I will show you some of the examples. You see the tremendous variability for the leaf. You see this is the standard and you get the mutations. Likewise this in the soybean color, the groundnut shape size, the seed number in the silakwa, and see the tremendous variability for the seed color in the black gram. And then this is the mustard. We have first institution in India or in the world, you can say, that we have isolated the yellow mustard. The importance of this, generally it has a high oil content compared to the parents, and because of the fiber content is less. Okay. You see these are the cowpea seeds. So tremendous. I worked on the sunflower also. This is the parent plant, and you see the variability which you get it. So with the mutations, you increase the variability. Okay. You see the flowers, this standard flower, and you see the these may not be useful for your yield point of view. But if you see the basic study, if you do it, you will get like Arabidopsis. You name the character, they have the mutations. And because they were having the contrasting character, mutant for the contrasting character, immediately they went to the gene level. Less and until now the CRISPR is what? You have the gene sequence in your hand, then you search for the mutations in the particular, like a T-link or the CRISPR, whatever. Okay. So the having, you see the tremendous variability for the wheat. This is the number of the siliquis per plant or the main shooting axis. Now you see this is the standard cultivated varieties in India. We have developed the lines which are very tall, which are long, having the more number of the seeds and much more number in the seeds, the per unit length of the siliqui. Okay. So this is the effect of mutations. This is very good, one of the very good mutations in the brassica. Generally the brassica leaf color is a dark green, you see. But we have the mutations which is the yellow, and when this yellow was crossed with this thing, we got a light yellow. The importance of this is, if you see the yield per plant, this particular plant, it is higher than the dark green and the parent plant. The reason behind this is the efficient utilization of the photosynthesis. Here there is a wastage. In the dark green there is a wastage, but here it is utilized. Here there is an underutilization. So, as I said, how to increase the per unit, increase the yield per unit area. So these are the some of the characters which will help you to increase the production per unit. This is our very good example that, as I said, the stem rust, this was the yellow rust. This is a comparatively new disease in India, And this was like a big threat for the wheat cultivation. But see, this is the left-hand side, it's a susceptible, and this you see the resistant. In the field, not a single spot of the yellow spores on this. So it and maintained the most of the characters, rather the plant type is also improved. So like this, you have to think of getting this is the Mumbin plant. See, this is a completely green and rest of the field is yellow. Okay. This is one of the dwarf mutations in the mustard. Again, I will say that, okay, this is the first time we are reporting that the dwarfs were not at all available in the Brassica germplasm all over the world, and we are the first within a 1 meter we got a seed yield as good as what are the tall varieties. So we need to tailor the plant that, okay, these are the plants, this is the plant character, which will rather better than the existing variety. Then there is a possibility to increase the yield. This is just academics purpose I am showing this. This is extreme dwarf. It is just 11 centimeter. If you look at the sunflower plant, which goes near about 1.5 to 1.7 up to 2 meter also. Okay, but it's just 11 centimeter. We got these mutations, but it was like academic use, not for the yield purpose. Similarly, in the soybean also. The tremendous variability for the plant height in the wheat. As I said, there is a biochemical character. Linseed is अलसी जिसको बोलते हैं. In the alsi seed or the linseed seed, there is a 50% of the linolenic acid. 50% linolenic acid because of the 3 double bonds. It gets degenerated fast, and because of that, the linseed oil is not at all edible. Okay, you get the rancidity fast, and generally this oil is used in the industry. But we have isolated a mutant which has reduced the linolenic acid from 50% to 2%. Now, because of that, what happens? Shelf life is decreased. Okay, and now it has decreased. The linolenic acid has decreased to the 2%. Shelf life, you can keep it for the longer period. Okay, so this is how the specific quality character can also be increased. These are some of the wheat mutant. Now everybody is aware about the health and what we want. Everything like a high iron. Everybody wants the high iron. So you eat the beetroot, then whatever red, there's a chili or the any other except red chilies. So you need the iron. So we have developed the mutations which is having the 42.7 compared to the 13.7, okay, milligram per kg. So there is a possibility, and you including G. This is one specific example of the sesamum. Sesamum all over the world or in India, its the size is, if you take 1000 seed and weigh, it becomes a 3.5 grams per 1000 seed. And this is not particularly in the confectionery industries, what we want is the boulder seed. Boulder seed is defined as when the 1000 seed weight of the sesame is more than 3.7 gram per 1000 seed. Then it is called as a board seed. But we have developed a mutant which is having the 4 gram and above. It is just released 1 month before Government of India notified this variety, and it will be popularized. Here I have forgotten to tell you this. The last line, if you see, the Indian Council of Agriculture Research tested this genotype of the seed, and which is less than 5% linolenic acid during the last year. So it is not our institution. BRC has not propagated. Government of India through Indian Council of Agriculture, we have the linseed line in our hand, which is now edible. This is the first variety you can say that you can use for the edible purposes. as an edible oil.
This is the same as flaxseed.
It's a flaxseed, not a kind of
But
edible.
flaxseed we have been eating for quite some time.
Yes, you can eat it, but if you extract the oil,
That will go rancid.
that also you can eat, but you cannot keep it for. See, my mother used to, we used to eat that linseed oil, but my mother used to say, you take this much amount, you go to the ghani and bring with the oil. So that is sufficient for the 1 month. So being a child, I used to always furious inside. Why can't she take one goni of the linseed and that will be useful for throughout the year? Or she used to emphasize, no, we'll do for this month only. And now I realize that why she was saying. Because it gets the rancidity, picks up the rancidity fast. Okay? Now with this climate change, there is uncontrolled rain. So if the crop is at the mature stage, like as I said, give an example of the soybean. So complete soybean, maximum area of the soybean, and farmers could not harvest a single part. Sometimes you read the situation of the farmer's suicide. I should not go for it. Rather, I suggest to the farmers, but this is not the right way. But when somebody takes the high amount of the loan, and she said, this time I couldn't get a single money, what to do? So they takes the wrong path, and therefore some of the situation created, whatever may be the agency was created, but the farmers are not able to do it. So when the crop is vanished, everything is finished for the farmers. So, but in the wheat, you find that if you take near about 10 years before this thing, the complete wheat crop in the Punjab and Haryana, it was under the water when it was about to harvest, and it has germinated. So with the mutations, again, we have isolated. Even though there is a water, it has fallen down, but it will not germinate. So if the farmer take a quick action, at least he can save something. Okay? So that is again the power of mutations. Using this power, we have developed near about 62 high-yielding varieties in the various crop plants, and our success stories are there in the groundnut. I will show you, followed by mustard, and in the pulse crops, it is like a moong, wheat, pigeon pea, and cow pea. And now we have made inroads for the linseed, sesame. Rice is now coming up, sorghum. So all these things, it is the importance of this is that okay, we are working since last 50 years, and after the 50 years, we have near about the 68 varieties. That if it is happens within the year or so, if it is 75, so you can say within the 50 years we have developed the 75. It goes along. And because of the paucity of the power in the BRC, that if it is a one crop, the only one person will work for everything. Agriculture is a big area, but starting from the research, your analysis, your extension, your trial, so everything one person. If you compare the kind of the manpower with the Indian Council of Agriculture Research, they have the crop-wise research institution. In the Gujarat itself only there is a groundnut research, NRCG, National Research Centre, with a big manpower, say 50 or the 60. And in our institution, one person, one crop. And he has to do all the things. Okay. So with that, I'm not saying that is a lacuna of our institute, but the manpower wise, but whatever results we got, sometimes we get a very high results in the particular year, sometimes it is very less. So it is a story of near about 50 years which we have come to this particular stage, and this is the graph. And some of the very good successful stories are there in the groundnut, bean, and in the oats. I will show you some of the pictorial. Now coming to the oil seeds.
So these have all been adopted by
Yeah,
farmers?
yeah, everything is over, they are with the farmers. Oil, it is now, it's a very important one. What is the amount of the expenditure government of India must be doing to get you the your French fries fried? What is the amount of expenditure? You know, it is a 75,200,000. That is the burden of India. That government of India makes the import of the oil worth rupees 80,000, nearby 80 to 90,000 crores rupees. And that we are using as a luxury by eating the French fries. Looking into the youngsters, I said, but that is being used in the house and everywhere. So it's not, it's a hidden work of the government. And there is no alternative for the government. Government of India cannot say that, okay, we'll not import the oil. We'll not import the oil, so you come down. So this is what there is a lot of. And therefore, why we are particularly concentrating on the oil seed agriculture, it is because of that. And day by day, the expenditure is increasing. When I started work on the oil seeds in 1986, it was a 10,000 rupees input, and now it is a 90,000 crores rupees. The reasons are, again, the area is more or less constant, and as well as the production is the constant. So therefore, whatever I was saying in the previous slide, that we need to increase the yield per unit area. And there are the ways, various ways of increasing the output by tailoring the particular plant. This success story, if you see, we have near about 16 varieties. And for the state of Gujarat, I will say, see, this is TG1. Then 1991, Somnath. Then I will come that 99 TG26. Okay. And then the TG37A. And now TAG24. This is for the all over the country now. The Prime Minister of the President of India, then President Mukherjee, has denoted or the devoted, nominated this variety on the India, saying that yes, this variety had changed the oilseed productions. So this is what it's for the almost all states we are working, and Gujarat state has particularly benefited. I will show some of these are the varieties, and these are the characteristics of the particular thing. TAG24, it's a very good variety. Sometimes he says that, you see the number. Yesterday I was with the farmer in the Mehsana district part of the village under the Seva she has introduced. And when I asked which variety you are cultivating, they said some of the name, and they said 24. 24. They were not knowing this is TAG24. But the ladies, which was not knowing anything about it, 24. 24 number ka variety. Then I said, this is from, then I called to the original person or the scientist who developed this variety, and he felt very happy. So there is a large number of the area in the Gujarat where our varieties are being prominently. Some of the characteristics, and this one I will say that whenever our varieties goes in the Gujarat market, and if you say that this is the tag variety, they nominated it as a tag variety. The moment you say it is a tag variety, tag stands for TAG. Okay? The moment you say this is the tag variety, the farmers will get 100 rupees more for the one. That is what the benefit to the. It's almost all states we have the good variety. Farmers are very happy. This is the that Gujarat Mandi, where the tag varieties are given the 100 rupees more per quintal. This is what our contribution. Now ICR is a big institution, ours is a very small, and I said one person works on that. But in the seed production, it is still up to 23, you can say this is the old data, okay, up to 19, but up to 23, now it has come to the 10%. Seed production means whatever seed is required by the farmers. So Government of India produces through the National Seed Corporation and the State Seed Corporation as well as the progressive farmers. So our contribution at the Government of India level is 10%. Okay, so we feel happy. Yes, we have done something for the. This is my varieties. As the Bhuvan, sometimes she overestimates me and says that whatever cultivation of the mustard in the India, it is mine. It is not like a mine, but I tried my best since last 25 years or so to develop some of the variety. And now 1, 2, 3, 4, 5, 6, 7, 8, and again 2 more. So 10 varieties developed in the recent years. Last year seed production was a 4% at the India level. So this I want to increase up to the 25%. So if it goes like that, I can proudly say that, okay, at least single person contribution at the India level, 25%. But I will have to wait for the another 5 years or the 7 years. Because the popularization of the varieties, like yesterday when I was talking, they said we want the seed. So the popularization process is a little bit slow because to cover the already, variety has to show its potential with the farmer, not what I said. Because I say, no, my variety is very good, you take it. And then next year farmer comes, and say it is not good variety. So I will be in the soup. It happened. The castor, they were showing number 7. Then in the Gujarat agriculture, they have given number 9 and number 9 and number 10. So the farmer rejected 9 and 10. We don't want this. We want only 7 because they found some good characters. Okay. This is very good field that whatever I talk, you don't believe on us. I will say what this person is lecturing and he say don't believe. Yes, you don't believe. This is the practical science. Agriculture is a practical science. Whatever technology you develop, you have to show your strength in the hospital. There is no option. You publish the paper, nobody will stop. Agriculture is a field, if you are developing the high yielding varieties, it has to show your strength in the farmer's field. So yesterday I felt so happy that among the 25-30 ladies, near 5-6 ladies in that area, they said, no, sir, cultivate the 24. That is what the honor for us. We don't want the honors, but saying some farmer that yes, I am cultivating your variety. This is what now in the Maharashtra there was no mustard cultivation, but the people have started, and you see the type of the plant and the amount of the silicoas it is coming, and there was a problem in the Maharashtra that stray animals, particularly from the forest as well as your domestic animals, they eat the other crops like a wheat you take, the chickpea you say, or any other crop they will eat, but they will not touch to the mustard. So the farmers which are in the border area of the forest, they started cultivating this, and they are very happy. At the same time, they get the good price in the market also, 7,000 to 8,000 rupees quintal per hectare, rupees per quintal. So they are very happy. And the development of the, Maharashtra is not suitable condition for the mustard, which previously we used to say. But if you 2 months of the winter, that is sufficient for the mustard to grow and to develop the seed at the proper size. So nowadays the farmers are interested to cultivate, and as I said that the 4% seed production is from our variety. So maybe in the coming years it will increase. This is the field demonstration with the all authorities of the state and the agriculture universities and the farmers. Which say that they are very happy, particularly one woman group in the particular area. They say, sir, in the Marathi, what you say, the आम्ही seed घेतला आणि फेकला. It means that, sir, whatever seed you have given, we have taken it and just thrown, thinking that it will not come. And the crop was like this. So they said it's a fantastic crop. We want to cultivate that one. This is another farmer, he has done sophisticated cultivation and the yield was very high. This is what that variety is, the linseed variety, where the oil percent is 36, oil yield, and this is the linoleic acid, which is in general, which is 50% in the control population, and it's very less in our. This is our pulse program. We are working on the Mumbai, cowpea, pigeon pea, and urd bean. And these are the varieties having the various characteristics like resistant to the various diseases. And these are the states and the year of release. Okay, we'll give some examples like here. TAU1. Okay, 90% of the urd bean cultivation, black gram cultivation in the Maharashtra, it is under this variety, or TAU1. So sometimes jokingly we say, suppose you are eating your idlis and sambar early in the morning as a breakfast in the Maharashtra, probably that might have come from our variety, because urd bean is one of the main components. So that is how, I will not go into the details of all these variety, but another variety which is of the, pigeon pea particularly.
Mr. Jambhulkar, I think, shall we take some questions because we are left with 15 minutes or so? Shall we be taking some questions from the audience?
Yeah. Now itself only, or?
Yeah. Right. So we have around 15-20 minutes left.
Or maybe within 5 minutes.
Yeah,
I will.
sure, okay.
So some of the important diseases like a powdery mildew, which if you go to the, you see the mung bean plant, which will become completely white. So then there is a completely loss of the yield. So most of our varieties are the disease resistant. This is the pigeon variety, TZT501, that first one. It is in the MP once we visited, and both sides of the kilometers together, it was like our variety. And when we used to ask which is this variety, the farmer used to say this is the number is your TJT50. You feel very happy looking into your crop, which is your child. It's like a flourishing in the farmer's field. This is the details of some of the varieties. This is the TAU1 I explained. These are some of the mung varieties resistant to pigeon pea disease. And this is the farmers in the Hyderabad or the Andhra Pradesh. So they have just cut the rice crop, and before that they have sown this mung variety. It's coming up like anything. This is again TMB 37. This is what the TJT variety, and the farmers in the, again, what is our contribution in the seed production? It is also near about the 10%. It's average if you take of all the pulse varieties, again it is around 10% contribution of ERC in the seed production. Recently we have released the wheat varieties this year, 1 month before we got notified, and the yield goes up to about experimental, it's 75 quintals per hectare. So even it goes near about 50 quintals in the farmer's field, still it is more than sufficient with the various disease resistance. This is another variety of the wheat which are released. And there is a lot of rice variety which are coming up. Not only that, whatever mutations or the varieties, we do the molecular characterization to publish the basic research, the change what happens in the DNA level, that also we do. This is another area of the application of the nuclear techniques or the nuclear energy to control the insect population. So irradiate the male insect, so that will become sterile and the population of the insect decreases down. But it requires the isolated area. That is more. What are the details of the cube? We have the tissue culture lab. So we do again the somoclonal variations or the isolate the desirable plant types through the mutations in that, and this is one of the dwarf variety which are isolated. It has not yet released, but having the all desirable characters. Okay, our authority generally they visit our field. If you see this particular case, he is Dr. Anil Kapoor, who was a director of the BRC, and afterwards he became a chairman of the Department of Atomic Energy, and he was the key person in the doing nuclear deal. Yes, that is 1, 2, 3 deal person. So our authority, our field. This is Dr. Sinha, who was a BRC director, after chairman, or various scientists visits this field and interacts with the farmers. And this is how we have covered more or less almost say India. See that farmers which are cultivating particular crop, our varieties or the seeds of so it comes. Then there is some kind of the basic research and the applied research all over the India. It is not only the BRC, so and in the world also there is a lot of work. And in the world, near about 2,450 mutant varieties has been developed. And this classification, the country-wise, where the China is in the top, followed by Japan and then India. These are the advances in the mutation breeding, and from the biology side, we must be doing what is just to say that this is the sterile insect technique which I told you. We have developed a kit for the testing of the organic carbon soil, to which you come to know, because the organic carbon, whether the soil is having the good fertility or not, you can decide on develop that also. This is one microbe called as a trichoderma. So our colleague has worked on that. He induced the mutations, and then that mutant has been formulated in some kind of for the soil treatment and now seed treatment, and we found that there is a lot of difference when you treat the seed with that particular microorganism. There is another plant is called as the Susbenia, which fixes the atmospheric nitrogen. So far you must be knowing that only underground nodules in the pulse crop, they fix the atmospheric nitrogen. This is a green matter crop which fixes the atmospheric nitrogen at the tune of, in the 55 days, it fixes the nitrogen of 120 kg per hectare. That is as good as urea. Continuous use of the fertilizers, our soil is getting deteriorated. Now there is a need, as the organic farming is coming up, and there is a large demand of the we need food. So, but if the soil health is not good, how to go ahead? So we have developed the which will take care of the soil. This is how we do it. The food irradiation is another topic where nuclear energy is being used to enhance the shelf life extension, to enhance the shelf life to remove the unwanted bacteria, insect pest, and it's a clean treatment. The atoms which you can take starting from the pulses, some spices, including jams or potato or the fishery product or the chicken or the meat. Anything is possible, and you see the after irradiation, you see the effect. And generally the tomato and the potato they get sprouts, but if you irradiate with the particular specific energy level, the spoilage is stopped. Okay. So this is how now the government of India is encouraging this, and you see the mangoes particularly, or the grapes, or the pomegranate. Suppose if you want to export it, in between, if you keep it like this, they will spoil. But if you irradiate it, their shelf life extend, and you will get the good remuneration out of that. You can do with the rice or the pulses, the chickpea. Previous one?
Questions can we take from audience?
Pardon?
Can we take questions from audience now?
Questions, no?
Yeah.
Yeah, I think there must be 4 or 5 slides only.
Okay.
It's finished now. Okay. So the insect disinfestation of the, and these are some of the agency at the national level and international level who approves the irradiation. So I am now in the end of my lecture that if really whatever we are eating, so one should think the farmer and is a visionary. This is the photograph out of the various crops, seeds of the various crops that we have presented in the Vigyan Bhavan. Back. The normal Borlaug says that you can't build a peaceful, you cannot build a peaceful world in the empty stomachs and the human misery. I think even I fully agree with this. It's very difficult. If you ask a hungry man to do the kind of the social work, he will say, no, I don't. First of all, you give me food. Otherwise, I will take the what will say the revenge of it. Okay. And the Buddha says that hunger is the greatest disease of the human being, and there is no treasure like this. And if we go to the World Health Organization data, the number of the persons suffering from the hunger and number of the persons suffering from the cancer, HIV, and the another 3 diseases, if you add up all these things, still you will find that the hungry peoples are more than the people suffering from the various disease. I was saying in the initial slide that everyone deserves the access of the healthy, affordable food and the quality nutrition care. And for that, nutritional food is or the crops is a very, very important. And the humble request to all of you is while eating, try to avoid this. You love the food and eat it. So that is one way to save the, even though one person wastes the 1 grain of the rice or the 1 grain of the wheat, and if you take a 142 crores of the people in this, so how much amount we are spending. If you go to the various party, the amount of the food we waste, it's really very, very high. So I request all of you to avoid the situation and enjoy the life. Thank you very much.
Questions?
Yeah, so I think it is exactly 1.5 hours. So I thought that whether I will be able to cover this much slides or not. But I did my job, and now, yeah.
It's a very basic question, but with your mutational breedings, you also mentioned that there were morphological changes like the plant height and such.
Yes.
So, have you observed that there were any changes or like pollinational changes?
Pollination.
Yes.
For the pollination.
Yes. Any changes in behavior of pollen?
No, we have not studied that. But see, it is an indicator the number of the seed sets on the particular plant. If your seed setting is good, depending on the habit of the pollination, it's a self-pollinated or the crop pollinated. If there is a no seed setting in the cross-pollinated crop, it means that there is a less pollination. And in the self-pollinated also, if it is a sterility, so there will be the less seed set. Okay, and whenever there is other insects, help them for the pollination. And if your seed setting is less, basically there is some. So such a plants where there is a less seed setting, as I said, that the unwanted plant we always reject, and only the desirable plant we select. So probably there must be some kind of the mutations in that, and that can be identified through the sterility. 100% sterility, that is good, so that we can use them in the hybrid seed production. But somewhere like intermediate, we always reject.
So while artificially mutating the plants or create by irradiation or various physical chemical methods, and picking the desired features, characteristics like the resistant, for example, resistant from particular disease, you may also, I'm not sure about that, but you may also select some features those are not much desired, and you may not directly identify it at the same time. So how would you deal with that?
Yeah, you are correct. As I given the example of one of our variety, that is a TPM1. Initially, it was selected for the yellow seed coat, but afterwards, when we analyzed, there is a high oil content. It is a disease resistant, and there is a change in the biochemistry, the reduction in the erucic acid. Likewise, in a one mutations, you are correct that there is a likelihood of changing the so many characters. Okay, but ultimately, when we go to the release level, we do all the nutritional aspects. Okay, all the nutritional aspects, and unless and until it is a desirable, then and then the proposal is accepted. Otherwise, not. Hopefully, even in that process, also we might have missed one, but it is not a detrimental. Say like oil seed. Oil is a main and fatty acid is a main. Okay. So if these two characters are the intact, though we have changed in some of the linseed, we have changed the fatty acid composition. In the erucic acid, in the mustard also we have changed. But we didn't find a single change which is harmful to the human being. We have the animal laboratories, so we do sometimes that study also whether it's harmful to the animal or not. Everything which is released is tested for the number of the characters, even for the number of the years and in the repeatedly. But you are correct that sometimes we don't know inside what is happening. Say like us, we sense something. The plant, though it is sense, but it don't tell. So you will have to analyze the situation.
So you had mentioned about physical and chemical mutagens. So considering that these are food crops, does using physical and chemical mutagens have like a side effect for human consumption, especially when you look at biological magnification?
Biological
Magnification.
magnification.
Yeah, consuming those crops does.
That is why I said. See, if you take an example of this room, we get the light. We get the light. But this rays, the sir will explain more nicely that these are the tolerable level. Okay? And when the energy level of this rays it increases, it's like a gamma rays, so it leads to the mutation. Now suppose the mutation has occurred. I was telling that what is the good points. Okay? There will be the changes. But whatever is good thing are there. But in the croplands we have seen as such there is no such a drastic biochemical changes which will lead to the kind of the cultivated plant to the poisonous plant. We have not seen such a thing. As I said, mutations are very, very harmful. But in the plant, there is a chance to select for the good one. Okay? Suppose something happens to me, okay? It's a way, see, it should not happen with anybody, but sometimes we do find the childs with the disabilities. But we cannot reject. It's our kit.
Plants have high tolerance level given their higher aneuploidy number of chromosomes.
Yeah, are you happy?
One more question. So you also mentioned about LD50.
Yeah.
Okay.
हाँ बोलो carry on.
We can have discussion after that. We will have a separate meeting. So I think
Thank you very much for your talk. It's a small token. Thank you.